sum up the nervous system by talking about these higher brain functions, going over some anatomy, and then the actual function. So here we go. I did include the lecture objectives, and you saw did you see the thing I sent out with the other two presentations that I'd forgotten to put objectives in it? Thank you, Tiana, for bringing that to my attention. So, not a problem. So this wiring diagram here is just a general overview of what we've been talking about and what we're going to mention today. We've got cortical function in the terms of motor activity through the corticospinal tract here, that's of course modulated through the input from the thalamus here, through the basal ganglia. Scaling is referred to the intensity of something. And then you get over here to the cerebellum that we're going to talk about today, which is involved in timing of movement. All of these things, of course, rely on proprioceptive input and then modulation through descending pathways, which you see, and there are many descending pathways, not just these two. But that's just kind of a general scheme of what we're talking about. When you're looking at the cerebellum, and we'll start with the cerebellum. The cerebellum essentially has, you can sum it up into three main components. One is equilibrium, because of the coordination between the vestibular nucleus and the cerebellum. The other is posturing. The cerebellum pays particular attention to the axial skeleton, whereas it also works with the extremities, but posturing is a key element of the cerebellum function. And finally, intentional movements are determined to a large extent by the cerebellum, not only in the movement itself, but in planning for movement. Planning for movement. Orientation to the cerebellum, it attaches to the back of the pons. We know that yesterday, primarily through the middle cerebellar peduncle. A couple of pieces and parts I've added here. There's the inferior colliculus, superior colliculus for orientation, cerebral aqueduct, fourth ventricle. This top portion here, the attachment, is called the superior medullary velum versus also the inferior medullary velum. The cerebellum is divided into gray matter and the white matter you see here centrally. We'll talk more about that later on. And then the cerebellum has essentially two cortices plus a midline structure called the vermis. You got, I said two cortices. It has two hemispheres plus the midline gray matter structure called the vermis. Now, I will tell you that if you go looking for the different components of the cerebellum, this is divided up into about 40 different components. But this is all you need to appreciate. You got two hemispheres and a vermis in the midline. I'm not going to get into it, but if you're on a neurology rotation, lesions of the vermis produce different results or produce different clinical symptoms than lesions of the cerebellum or other structures. So that's why I made the distinction there. So if you look at the cerebellum sort of structurally, you've got the pons right here sending its fibers, the pontocerebellar fibers, through the middle cerebellar peduncle here. Those fibers are distributing. You see the huge white matter mass right there. The central white matter mass of the cerebellum is right there. These fibers are headed out to the cortex of the cerebellum out there. The cortex projects to the predominant cell type of the cortex is called a Purkinje neuron. I'll talk about it in just a second. But it projects not out of the cerebellum, but it projects to cerebellar nuclei. They're called the deep cerebellar nuclei. If you dissect away the cerebellum here, you cut a hunk of it out, and you'll see these nuclear groups inside the cerebellum here. These are the deep cerebellar nuclei. It's the deep cerebellar nuclei that project out of the cerebellum.
When you keep saying project, do you mean it controls it or it leads to it?
It's provides input. The afferent input to the deep cerebellar nucleus would be the cerebellar gray matter. You understand what I just said? The cerebellar gray matter, the Purkinje neurons, their efferent projection is to the deep cerebellar nuclei. If you're a deep cerebellar nucleus, it receives an afferent projection from the cerebellar cortex or from the Purkinje neurons. It depends on your reference point. Right? You follow me? Kim, you still look confused. Okay. If the reference point is here, and I say, what is the efferent projection? That means, what is this projecting to? Remember anatomy? Afferent is toward the spinal cord, efferent is away. So what is the efferent projection of this? It's to that. If the reference point is this, and I say, what is the efferent projection of that? It's wherever it's going. But what is the afferent input to this? It's this. So it's your reference point. So in relation to the cerebellum, the Purkinje cells of the cortex project to or are efferent to the deep cerebellar nuclei. The deep cerebellar nuclei are efferent to the rest of the nervous system. Okay.
Is cortex synonymous with gray matter?
Yes, and the next slide is going to show that, or next couple of slides. Okay?
Did you say that the fibers of the pons distribute to the cortex?
Yes.
Okay.
The fibers, the predominant input to the cerebellum is via the pons. What's the predominant input to the pons? The basis pontis. What's the predominant input? The cerebral cortex. You've got the cerebral peduncle, right? The cerebral peduncle is here. The pyramidal tract, the pyramid, is down here. The predominant input that makes this big hump is the descending motor activity from the cortex. Okay? All right. So if you look at a cross-section through here, you have the deep cerebellum. There are 5 different deep cerebellar nuclei. I don't care that you know the names of these nuclei. Just know that there are deep cerebellar nuclei, and they provide the outflow or the efferent projections from the cerebellum. All right. Now let's look at the cortex. These little dudes here are called folia. They don't have gyri and sulci like we're going to see in the cerebral cortex. These little things are called folia. If we look at a folium, what you have is gray matter outside of white matter. If you look at this histologically, the gray matter is from here up, and there's the white matter in the center. The gray matter is divided into 3 different layers of the cerebellum, and it's consistent pretty much all through the cerebellum. This whole cerebellum pretty much has this structure. You have an outermost molecular layer, you see in this higher magnification here. The molecular layer contains the dendrites of the Purkinje neurons. as well as a whole lot of communication between the Purkinje neurons. The molecular layer, Purkinje layer, and then here is the granular layer. The granular layer contains billions and billions and billions of interneurons that make up the internal circuitry of the cerebellum. So those are the three layers of the gray matter of the cerebral cortex, cerebellar cortex, which surrounds the white matter. If you look at this, I told you the other day that the dendritic tree of the Purkinje fibers was ginormous, and here it is. This is the little axon, the one axon, and you can see this monstrous dendritic tree. They're arranged in a particular way. So if we look at it on FOSS, it looks like this. If you turn it 90 degrees, they line up like that. So they line up like this, oriented to the fovea. So if this is the ridge of the fovea right here, you've got these Purkinje neurons that are lined up like that. If you look at the circuitry, the internal circuitry in the inputs to the Purkinje neurons, there are two primary inputs. One of them, and we've talked about these, one of them is the climbing fibers. These come from the inferior olivary nucleus. You remember in the, when we were talking about the medulla, you have the pyramids right there, and then beside it you have the olives. The pre-olivary sulcus is where you'll find cranial nerve 12. The post-olivary sulcus is where you'll find cranial nerve 9 through 11. If you look at a cross-section of the medulla, you'll see the pyramids are right here. And then you've got this thing that looks like that. That's the inferior olive. Draw one of those pictures. So the climbing fibers come exclusively from the inferior olive. The red nucleus, remember the red nucleus facilitates flexion, projects to the inferior olive. That projects to the cerebellum. So that's how you get cerebellar control of some of the flexors right there, okay? That's how it's sort of wired up. Everything else that has input to the cerebellum is a mossy fiber. So when we were talking about, I mentioned the spinocerebellar pathway, the spinocerebellar tract, that's a mossy fiber. When we were talking just now about the pons projecting to the cerebellum through the middle cerebellar peduncle, those are all mossy fibers. If you look at the synaptic arrangement of things in the cerebellum, the input, the climbing fiber and the mossy fiber input to the Purkinje neurons is excitatory. Now there's another fiber, there's another cell type, it's called the parallel fibers. These are an internal cell, a neuron within the cerebellum that connect Purkinje neurons in a row. The parallel fibers are the ones that help connect the Purkinje neurons. They're excitatory too. Everything else, in terms of the white matter, is inhibitory, which means that the Purkinje neurons themselves inhibit the deep nuclei. This says it all right here. Those three are excitatory, everything else is inhibitory. okay? You don't need to know these names of these cells, you don't need to know which, these little pluses and minuses. All you need to know is that. Climbing fibers, mossy fibers, parallel fibers are excitatory. They excite the cerebellum. If the cerebellum is excited, the output is inhibitory. That's the bottom line. Now, we're talking about these parallel fibers, we talk about the mossy fibers and the climbing fibers. Understand that if you have the folia here, and you've got all of these Purkinje fibers with their dendritic trees arranged like this, the input to the cerebellar cortex, the gray matter, is in these parallel runs. So if I are going to move something, the Purkinje fibers that are involved in this are in a run like this, and they're all kind of connected. The cerebellum is very site-specific. It's not, you don't just get information just goes all over the place in the cerebellum. It's very site-specific. Fiber beams is how it's described here. Beams of related Purkinje neurons for a specific function. And you can summarize that this way. Cerebellar inputs have place-specific information. Place on your body. These runs result in burst of activity from the Purkinje fibers that are timed. And by time, you remember we were talking about summation the other day? Temporal and spatial summation, that sort of thing? You shook your head no. You don't remember the climbing and mossy fiber input? How you have one fiber that makes 1,000 inputs versus one other fiber type that will have one input but a whole bunch of Purkinje neurons. Go back and review that. You're going to probably see it again in multiple choice format in a few days. You might want to review it. Okay? So what happens is, and I mentioned that in order to excite one of these huge Purkinje neurons, you have to have an adequate input. That's why the output of the cerebellum is not constant. You got to get enough activity going to those Purkinje neurons to get them excited enough to fire. So it's timed burst of inhibition. Why do I say inhibition? Because I said that the Purkinje neurons are inhibitory. And what that does is it translates place on your body in time to create appropriate timing of muscle movement. Muscle movement. I said cerebellum is intention tremor muscle movement. Okay? What happens when it doesn't work? We talked about intention tremors the other day. On the physical exam, you're going to learn past pointing. So in order to test cerebellar function, basically all you have to do is put the body in motion. Put the body in motion. One of the best ways to do it is, Kayla, would you touch my finger with your finger? Now touch your nose. Back and forth. Back and forth. Back and forth. Back and forth. Now do it with the other hand. To do that, you have to have a well-functioning cerebellum. If you don't, do it to me. Let me do it to you. What will happen is, if you move your finger, you can't find your nose and you can't find her finger past pointing. This diodokinesis is the inability to perform rapid movements, get the body in motion. One of the, the way I do it, the way I test it, is I have the patient sit here and I test cerebellar function in the upper extremities and lower extremities. So I say, put your palms on your thighs. Now turn your hands over and back and do it as fast as you can. That takes an intact cerebellum. If I can't do that, that's the problem. For lower extremity, I say take one heel and run it down the other shin, like that. That's a purposeful movement that has to be coordinated. You can also have them tap the stool of the table. And, I get them to stand up and hop on one foot, hop on the other foot, and things like that. So those are tests of cerebellar function. Nystagmus involves an cerebellar dysfunction related to the extraocular eye movement muscles. Nystagmus. And this is what I want you to remember here, that the largest input to the cerebellum comes through the basis pontis from the cerebral cortex. Now, understand that the cerebral cortex projects to the same side pons, because the crossing doesn't occur until the pyramidal decussation. So this says right here to the contralateral, to the opposite cerebrum, where is the crossing? The crossing has to be in the pons. So the cerebral cortex, the motor strip, area 4, projects to the basis pontis. The fibers go across to the other side to go through the middle cerebral pedicle, cerebellar pedicle on the other side. Okay? The crossing occurs in the pons. Lower down, the corticospinal tract crosses at the pyramidal decussation. Okay.
So then, whether we're talking about corticospinal or other like extrapyramidal tracts, they're all going to start in that motor precentral gyrus. I guess I'm very confused about extrapyramidal and pyramidal.
Okay, so let's just call the corticospinal, what's going to form the corticospinal, that's pyramidal. It originates from a pyramid neuron up here. Everything else is extrapyramidal, particularly the basal ganglia, the substantia nigra, okay, the cerebellum, because it's not corticospinal. All right?
Do the corticospinal tracts have to go through the cerebellum as well?
No. The corticospinal tract is let me say it this way. The corticospinal tract is a straight shot from here to the spinal cord. But that straight shot has to be modulated in 1,000 different places. One of the places that it's modulated is if I'm going to move, I send all of that input downward. It gets into the cerebellum. The output of the cerebellum eventually makes its way back to the cortex to refine how much of the movement is actually going to occur. I mean, that's the way you have to think of it, is that the basal ganglia, the substantia nigra, the cerebellum, they're modulating movement. They modulate what actually ends up in the spinal cord. Okay? Did that help, or is that, you're still confused?
So my understanding then is there's a connection between there, but the actual movement, the final thing that leaves is just corticospinal.
Yes.
But before
the
you-
final
before you move it,
the
modulation
final
occurs.
movement is corticospinal. But in order to make that movement what it is, it has to be refined. You know, like I stepped off of the step the other day and I said, why is it that I don't fall? Why did I not jump up in the air? All of these other systems, all of these extrapyramidal systems are what makes me not jump in the air. It's refined movement. And that's what this is. Okay? All right. Let's move to the cerebral cortex. The cerebral cortex, like the cerebellum, has gray matter on top of the white matter. These little holes in here, this is not spongiform encephalopathy. They're blood vessels that have no blood in them. So this is the white matter down here. Just to make you aware of this, and I want you to pay attention to this, this goes all the way back to anatomy. And I mentioned it earlier when we were talking about, sometime this week, the main vascular supply of the brain comes from the internal carotids and the vertebrals. They form the circle of Willis with three main arteries. The anterior cerebral artery supplies the medial part of the cortex. The middle cerebral artery supplies the dorsolateral part of the cortex. And the occipital artery supplies the bottom part of the temporal lobe, but primarily the visual cortex back here. You need to keep that in mind. You will see that in multiple choice format later on.
We say that one more time.
Yeah, okay. Going back to anatomy. Remember the anterior cerebral arteries, they come off of the circle of Willis and go up and over the corpus callosum. That's the corpus callosum right there. So if you're going to find the anterior cerebral arteries, you open up the fissure and there they are down there. They supply the medial part of the cortex. The middle cerebral arteries come off of the circle of Willis and go up through the lateral fissure here and supply the dorsolateral cortex out there. And then essentially the posterior cerebral supplies the visual cortex. We'll just say the visual cortex. Okay? There are three types of cortex based on their structure, based on their layering. The archicortex is the oldest cortex because it only has three distinct layers. And when I talk about layers, these are three different stains. And neurophysiologists and neuroanatomists have somehow decided that there are 6 layers in the most developed type of cortex. But if you look at the archicortex, it's only 3 layers, which would be, if you go back to the limbic system involves primitive behaviors, which is phylogenetically the oldest cortex. So if you happen to be a newt, their entire cortex is nothing but 3 layers. You're only going to find the most advanced cortex, the neocortex, which constitutes most of our cortex, in primates. That may not exactly be true. Lower primates like deer and dogs and stuff, they may have neocortex, but it's much less of it. Okay? This is the phylogenetically newest cortex, and in between those is the paleocortex, which involves older systems like olfaction. Thankfully, most of our cortex is neocortex. The predominant cell type in the cerebral cortex is the pyramidal cell. There are many different cell types in the cortex. You don't need to memorize these cells. But the predominant outflow of the cortex is going to be through the pyramidal cell that you see here. Fusiform cells also pass out of the cortex, but the predominant cell type is the pyramidal. There are neurotransmitters that predominate in the cerebral cortex. Glutamate, which is an amino acid, essentially, it is, but glutamate is an excitatory neurotransmitter. GABA, if you have inhibition, is the predominant inhibitory neurotransmitter that you see in the cortex. You remember Brodmann's areas that I started to discuss yesterday, I believe, 40-something different areas. The ones that are significant, we're going to talk a little bit about today, are area that some of you already know. Area 312 is the primary sensory area, which is in the post-central gyrus. Area 4 is the primary motor area. Contributing to 4 is area 6 and 8. These are called the frontal eye fields and the premotor. What these things are is area 4, the premotor cortex, the primary motor strip, that's where when information leaves there, it's headed to the corticospinal system. But the initiation of movement starts elsewhere, like in areas 6 and 8. So if I'm sitting here and I said, I want to pick up that Greek yogurt cup, the desire to initiate movement comes in 6 and 8. They start the ball rolling, and it's area 4 that is the primary movement area. We have, I mentioned yesterday, areas 17, 18, and 19 is the primary and associational vision areas. The cingulate gyrus, we're going to talk about at the end of the day, is here. Cingulate gyrus. The auditory area that was on a slide from the brainstem lecture is right here. That's in the temporal lobe. It's also called the and you may hear this sometime. It's called the transverse gyrus of Heschel. H-E-S-C-H-L. Heschel. The transverse gyrus of Heschel is in the temporal lobe. You don't need to know that. I think it's just a cool word. Heschel, Heschel, Heschel. That's cool. Okay. The two other ones we're going to talk about today are areas 22 and 44 and 45. Those would be Wernicke's area and Broca's area, and they are involved in speech. All right. On this slide right here, there's a lot of stuff going on this slide, but this is what I want you to pay attention to here. The afferent fibers, or the afferent projections to the cerebral cortex, come from 3 primary areas. You see where it says cortical association? This means parts of the cortex talk to other parts of the cortex. Callosal means that one hemisphere talks to the other hemisphere. And thalamocortical means what we were talking about yesterday. The thalamus projects to the cortex. That's the afferent input. The output from the cerebellum is over here. Association and callosal. So from one part of the hemisphere to another part of the hemisphere. Callosal, one hemisphere to the other. Corticospinal. Corticofugal. Fugal means just projects. So the cortex projects to other areas. The cortex would project to the red nucleus. any projection fiber other than the corticospinal and the corticothalamic. If you look at neurotransmitter systems in the cortex and their influence on the cortex, you have a large dopaminergic projection to the cerebral cortex from the mesocephalon area here. We know that substantia nigra to the caudate movement. You get too little, you got Parkinson's. You got too much dopamine, you have schizophrenia. And that's a very general statement. Noradrenergic pathways, a lot of learning, behavior, memory comes from these norepinephrine neurotransmitter systems. Wakefulness comes from noradrenergic. So when you're asleep, how do you wake up? What wakes up the brain? How do you wake up? What starts the process of waking up? It's that little dude right there. Boink! It's a nucleus. I didn't even mention it when we were talking about the brainstem. Just for you, if you're on Jeopardy, it's called the locus ceruleus. That's kind of a cool name, ceruleus, kind of like a Roman, not spelled that way anyway. That's what wakes up the brain. It's a noradrenergic system. Serotonergic pathways, the big deal about serotonergic pathways is their involvement in mood. And we talked about SSRIs, about increasing the amount of serotonin in the brain as a treatment for depression. And finally, cholinergic pathways, acetylcholine is a huge player in the cortex, particularly in terms of memory, learning and memory. Because if you don't have learning and memory, what you have is dementia. If you can't remember what you ate this morning, or you can't remember what happened 5 minutes ago, that's dementia. The guy I was telling you about that I take care of, dementia, that's the thing. I told you, you ask him something, 2 seconds later, the same thing, you're in a circle. Alzheimer's dementia, you treat it with drugs to increase acetylcholine. Now, how does the cortex work? Interestingly, you've got these 6 layers, right? All of the neurons that are in these 6 layers are tied in together. You remember we were talking about the beams, the parallel beams in the cerebellum run this way? The cortex is similarly connected, but this way. They're called cortical columns. So what they've done in this experiment here is they've injected a radioactive tracer into one eyeball of an animal, and they stimulated the vision of that eyeball and picked up where the tracer went. And it turns out that the projections, or the cortical neurons that are excited by that one eyeball are like this, in these cortical columns. And they define these cortical columns so that if you're looking at this spot in space, you're going to activate these cortical columns. Area 17, the primary visual area, looks like this. So every spot in your visual field has some cortical columns that are associated with it. The reason there are gaps in here is because there's a difference between visual fields like up and below, temporal and nasal. This is just one visual field. If you were to stimulate the entire visual field, you'd have a complete pattern here. The Nobel Prize was awarded for the discovery of cortical columns, and that's how most of your nervous system, your cortex, works. Whether you're talking about vision, motor, whatever, the neurons that are in that column of white matter, excuse me, gray matter, are arranged this way. Let me give you one example here that's really cool. All right, so vision, one of the relays for vision is the lateral geniculate, right? Vision goes through the thalamus. The nucleus in the thalamus that controls vision is the lateral nucleus, right? So here's my visual fields right here, my two eyeballs. So here in the retina, here. We know that the retinal projections are like that, right? Everybody follow me? This would be the optic chiasm right here. So this is going to go back to this lateral geniculate. Remember I said yesterday, the left lateral geniculate is going to supply this retina and that retina, which sees that visual field. So the left geniculate sees the right visual field and vice versa. Okay? If you take a section through the lateral geniculate, it actually is arranged in 6 layers. If you pick a point in the visual field, right there, you'll see that and find those neurons that are activated in the geniculate here, you'll see that these cells that see that point light up in certain columns in certain lamina from this eyeball. From the other eyeball, which sees the same point in space, this column lights up here. Is that not cool? That, you're not going to be testing on it, but that column of neurons seeing 2 different areas is consistent between crossed and uncrossed fibers of the optic tract. That's just cool. It's like Etchel. You just like to do it. If you look at the projections to the cortex, different stimuli, this is what you see. If you're passively viewing words, you're not reading, you're not saying them aloud, and you do PET scans, which light up the area of the cortex involved in that activity, you'll see, if you're just looking at them, you'll see that you get a lot of stuff going on out here in the visual cortex. If you listen to words, then you're going to see the transverse gyrus of Heschel lights up, the primary auditory area. If you're speaking words, you're going to do the primary speech center. If you're generating verbs, in other words, if you're stringing words together into a statement, then you light up another area. Wernicke's area. Right. So you can map how the brain works through PET scanning like that. Now, here's the thing you got to watch. You got to pay attention to. And this is kind of like the touchy-feely part of this lecture. Then we'll take a break. Everything about your understanding, sensation, being, affect, who you're sitting next to, what you're sitting on, everything is part of the nervous system. Which is interesting, because when you go and test the nervous system in your phys diet, you test in 2 different ways. The first part is you test the brain. In other words, the connectivity, pathways, being able to sense, being able to move. That's the brain. That's the hardwiring. But then you also have to look at the mind, which would be doing a mental status exam to determine your cognition and other things that make you you. So unlike the heart, I mean, when you're examining the heart, all you do, you examine the mechanical beating of the heart. It's boring. It's only one thing that you look at. Is the heart functioning or not? Here, you have two different elements. One is the hard wiring, the other is the psych component. Have you guys heard of the Folstein Mini Mental Status Exam? Okay, that's the mind part. Doing this, that's the brain part. All right? I just throw this in here. These are kind of interesting things in terms of the mind aspect of things. Especially this part right here, spirituality and mysticism. These are uniquely human things. Okay? Go take a break and then we'll come back.